Okay, so now we're into DNA and RNA.
Where are we?
Page 23. Okay, this stuff is a little bit more difficult. We went over this in the heights of Corona. I was quarantined, y'all were quarantined, so if you forget some of this stuff, it's okay. But I'm telling you right now, there's always a DNA question on your STAR test. There's always an RNA question, and they always relate DNA and RNA and other things. So we need to figure out what's going on, okay? So, the blank of the cell contains all of the genetic information in the cell. What organelle? The blank contains the genetic information.
Nucleus.
Nucleus. Okay, contains the genetic information. For the most part, it's DNA. Okay, so the nucleus has the DNA. But we did learn from the nucleic acids, it could be R, ribonucleic acid, or D, deoxyribonucleic acid, DNA or an RNA. Would you like to borrow one of my pencils?
Sure.
Okay, come here. Here's a good one. You can just give it back to me. Okay, so first off, we need to know what the monomer, mono meaning one, monomer of a nucleic acid is, and the polymer, polymany. So guys, if this is a DNA strand, so remember it has the backbone, and normally DNA strands are twisted, but I'm just going to straighten it out for ease purposes. Okay, so here's your DNA strand. The building block of this is a little bit of the outside and a little bit of the inside, right? DNA is not just made up of the outside backbone or the inside steps. So we're going to learn that this is a base connected to a sugar and then connected to a phosphate. So this right here is the monomer, the building block, and this is called a nucleotide. Okay, so here's the nucleotide, which is a sugar, a phosphate, and a base. Now remember, we said, now this is flipped upside down, but I think of the Superman logo on his chest. Superman, okay, so this is the sugar. And what is the chemical compound of sugar? C6H12O6. We have a phosphate, which contains phosphorus. And we have nitrogenous bases that contain nitrogen. So when we talk about the elements in a nucleic acid, such as DNA, chomp, the cho is in the sugar, the nitrogen is in the nitrogenous base, and the phosphorus is in the phosphate. So that's how we have all of the elements of a nucleic acid. Okay, so this is how they base pair in DNA. Apples in the tree, cars in the garage, okay? And this is just when it's in the nucleus, okay? So remember, when we're in the nucleus and we have DNA, A bonds to T, T bonds to A. G bonds to C, C bonds to G. What?
Are we going to do that genetics with that SpongeBob stuff again?
Maybe a little bit, yeah.
That was hard.
Yeah. Okay, so up here, help me finish this. You may do it at your seat or you could do it with me. So it says, write out the DNA base sequence that will be complementary with the following. So guys, complementary means it goes with it. It's not the same, it goes with it. So what goes with the A? T, A, T, G, C, G. Guys, good, you got it. Finish the rest of them. I'm going to come around, check that last one is spelled out correctly. Apples in the tree, cars in the garage. Now, this is only with DNA in the nucleus. Yep. Yep. No. Good. Good. Okay. Okay. So up here, All of the following are found in a DNA molecule. So DNA is a nucleic acid, cho-cho-chon chomp. Okay, so is carbon dioxide in a DNA? Is a sugar? Is a nitrogen? Is a phosphate? Yes. So there is no carbon dioxide in DNA. There's carbon and oxygen, but there's not a CO2, not a carbon dioxide. Okay. Before we get to the next part, which is DNA to RNA, I just want to go over. Oh, sorry guys, we were learning about writing checks for a project in IPC. Okay, so if this is a big old animal cell, this is as round as I'm going to get it, and we have our nucleus, and I'm putting the nucleus over to the side just so I could do stuff outside the nucleus. Remember, the nucleus has pores, nuclear pores, and things can go in and out. Now, DNA cannot go in and out because it is too big and bulky. DNA is in its double helix form, and it's just wound up really tight, can't fit out the holes, okay? The only place DNA is ever is in the nucleus. Now, unless it's in a bacteria, because bacteria don't have a nucleus, they're a prokaryote, but for the most part, STAR test is going to ask you about your eukaryotic cells, okay? DNA cannot go out. Oh no, so DNA is our master copy. It has all the instructions for all the proteins we need. But the ribosome, remember it kind of looks like a hamburger bun, is out here in the cytoplasm, and this is actually what makes the protein. So we need to get the message that is in the DNA to the ribosome. Who delivers that message? Messenger RNA. Good. So what happens is DNA is made up of A, T, G, C. It's double, meaning it has two sides, and it's always in the nucleus. So what happens is DNA opens, but instead RNA bases come in. Now RNA will be single, and it's apples under the tree, cars in the garage. So say if this sequence is A, T, G, C, what is the RNA complementary? What bonds with A?
U.
A to the U, apples under, good. What bonds with the T?
A.
A, good. What bonds with the G? C. What bonds with the C? G, good. So now this smaller sequence can fit out the nucleus and find a ribosome all the way over here. And the ribosome reads it 3 bases at a time, okay? So we needed to turn DNA into RNA, and then the ribosome's going to read it 3 bases at a time, which is called a codon, okay? And then remember we had to use that chart to figure out which amino acids the codon coded for. So look up here. Every 3 bases calls in an amino acid, and then this next one calls in another amino acid, then another amino acid. Then the amino acid chain folds into the protein. So how do we get a protein? It goes DNA to RNA to protein. Okay? Now, technically the process, DNA turning into RNA, is called transcription. RNA to protein is called translation. And what did the Amoeba Sister video said that says the blank comes before the blank? The C comes before the L. If you forget which is which, transcription comes before translation. Okay? So let's see if that helps us with our worksheet. Okay. The process passing information from DNA to RNA. What did we just say it was called? Trans-DNA to RNA.
Encryption.
With a C. Trans-secryption. That comes first. DNA pairs with RNA. The only change is that T is now replaced by U. Apple's under. So it's U bonded to A, C is still with G. Okay? So now, let's do the first one together and then y'all finish. Ready? Apples under. Apples under. Tree. Good. Yeah. G. Apples. Apples. Good. Okay, finish the rest too. I'm going to come around and check. So like succession, natural selection, that took like a couple days of our time. This DNA to RNA protein, that took like a month. So that's why it feels like we didn't accomplish a lot, but we really did.
Didn't it get kind of spread out because of COVID?
Mm-hmm. And just a reminder, I have on here DNA versus RNA. So we got our DNA bases, RNA bases, transcription, translation. I might add a little bit more, especially if I make this front back. But I've got a little segment here on our review sheet on that. Okay. So, now the process from RNA to amino acids, and technically amino acids are proteins, so RNA to protein is translation. Good. With an L. RNA is read every 3 bases called a codon. Every codon codes for an amino acid. Remember, there's a start codon, and then there's a end codon. Okay. This we know, so like the STAR test also gives you a lot of pictures, and based on the pictures you could tell what the answer is. If they give you a picture of this right here, this is DNA. Why? Why do we know that's DNA?
Because it's a double strand.
Double? Why else?
It has T's.
It has T's, good. And if it were in the nucleus, you'd be like, oh yeah, that's the nucleus. Okay, why is this definitely RNA or messenger RNA? Single, and then I heard somebody say U, good. And why do we know this is amino acids or proteins? You can actually read methionine, isoleucine, serine. So these are definitely amino acids. So we know that's a protein. Okay? Awesome. I want to get to the codon chart. I thought I gave, okay, here it is.
Heroin.
Turn to page 29 real quick. Okay, so remember, if I give you bases, and do you notice there are no T's on this chart? So if you got a codon that had a T in it, that's the DNA. You can only put messenger RNA in here. So if you got, for example, if you got ATG, you could not put that in there. What would you have to turn that into?
RNA.
And what is it?
UAC.
Good, awesome. Okay, so if I gave you A, first space goes here. Remember, then we stay in that column. We go over to the second one, and then we pick whichever the third one is. So we go A to the A to the A. First space, start with the A. Go over to the A. Tell me when to stop.
Lysine.
And then A is the, not the first, not the second, third. Good. Let's do another one. G, A, C. We start with the G, okay? First space G. Go over to the A. And then C is the second Glycine, good.
Glycine.
But you
now
went
one
too
over.
far. You're supposed to stop at the A.
Oh, thank you.
So,
G, oh, here it is. The A to the C. Thank you. Reading it up close actually is hard. Okay, let's do C-U-A. Start with the C, over to the U, A. Okay, this is what I want to show y'all. Look up here. Okay, this right here, phenylalanine, is UUC. UUC. If there was a mutation and it turned into UUU, it's still phenylalanine. So, this is what the STAR test is going to say. If it changes it to the exact same amino acid, will there be any difference in the protein? No. So, a 1 base mutation might be okay. Let's do another one. Ready? UUC. UUC. I want to mutate it to UUA. UUA. Uh-oh. Leucine. 1 amino acid difference still might be bad. Okay? Let's do another one. Ready? Watch this one. UAC. U to the A to the C. UAC. Tyrosine. What about UAA? Stop. So guys, if you ever change it to a stop, usually it changes the sequence of more than one, because what happens is, look up here, if you have an amino acid sequence and say this one changes to stop, then your protein only looks like this one because it stops. So you change the sequence of more than one amino acid. Okay, so I'm trying to think if there's anything else. Nope, because then we're going to move on to genetics. Okay, I'm going to give you 15 minutes to do, to see how far as you can get. Oh yeah, karyotypes, real quick. Karyotypes. You crack open a cell, you take all of your 46 human chromosomes and pair them up. You get 1 from mom, 1 from dad, 1 from mom, 1 from dad. So you have 23 pairs. Remember, you get a 1 from your mom, 1 from your dad. 1 from Bob, 1 from dad. Okay, anytime you see an extra or a missing, something is wrong. But I do want to show you the X and the Y chromosomes are allowed to be different because the pair 23 is your sex chromosomes. Okay? What does XY mean, boy or girl?
I do not know the answer.
Okay, so here's a hint. What are you always going to get from your mom? A girl, right? Chromosome, right?
Why?
Okay. And then the Y is from your boy. What kind? A Y. What does that look like? What part of the boy does that look like? See what I'm saying? Y is the boy. Y is for the boy. Okay. So guys, a boy, remember, the dad can determine if it's the female or the male because girls only have X's and X's. So a mom is only ever going to give an X. Okay. A boy can give an X or a Y. If he gives an X, it's a girl. If he gives a Y, it's a boy. Okay. Real quick, this is a short section of messenger RNA. Look up here. I knew this is RNA because it has a U in it. Which DNA code probably produced this? So are they asking for DNA or RNA? DNA. Make sure you understand what they're asking. What does it say? Mutations can happen. Oh, real quick, mutations up here. Mutations can happen in any cell, but only mutations that occur in gamete cells, sperm and egg, are passed on. So guys, if you have skin cancer, a mutation in your skin, are you going to pass it on to your baby? No, because it's not in your egg or sperm. So only mutations that occur in eggs and sperm actually get passed down. Okay, I'm going to let you go.